Hello and welcome to my class. This is chapter 14 of BIO101 Mendelian genetics. When we are talking about this topic, the person that we must first pay our respects to is Gregor Mendel. Now, Gregor Mendel is called the father of genetics in this modern day world, and he was an Austrian monk. Okay, so in those days, it was like an easy thing for very poor people to send one person in the family into the priesthood so that they have a better chance in life, like they have a chance at a better amount of education than the hard work in the fields or to be under a service person and so on and so forth. So Gregor Mendel here came from a poor family and was admitted into the monk to being a monk in the monastery in Austria. Okay, so a few topics that you should definitely know about this chapter are listed out in this slide and in the next. Okay. So we start with what was the general mode of thinking before Mendel and after Mendel. So we would start with that. So initially it was thought that like around in the 1800s, to explain heredity, people would say that it is kind of blending hypothesis was the idea at that time. That was the raging idea. And that meant characteristics of any offspring is intermediate between those of the parents, meaning it kind of mixed the characteristic of the parents, kind of got mixed like into a melting pot, and then you had your offsprings. But the downside of this was, how do you explain that you have different kinds of offspring? The same parents would have different kinds of offspring. If it was blending, it would mean to say that everyone would kind of come chock-a-block exactly like a blended mixture of the parents. But that wasn't really the case. Like even siblings, if you see, would have some characteristics of the mother and some of the father, and the other sibling might not have in the same way. And then later on, particulate hypothesis became more acceptable. It was offered as an alternative explanation. What they said was each quality or each characteristic is put into a particle. See, mind you, during Mendel's time, he did not know what is the gene or what is DNA. He did not know that. He just thought there were some particles that were present and that was being passed on for different generations. Okay, so Gregor Mendel came up with a set of experiments which helped lay the foundation to understand the way different characters are passed down from one generation to the other. Okay, so first a few terms that we have to surely know and understand and not get mixed up. Okay, a heritable feature. Heritable feature is what you inherit. Okay, inherit from your parents. Okay, so a character. Character meaning it can be hair color or hair quality or say eye color or say height or any of these different qualities you inherit from your parents, and that is a character. A trait, on the other hand, is a specific kind in the character, meaning black hair. This would just mean hair color is a character, but black hair is a trait. Eye color is a character. Green eyes is a trait. Height is a character. Being tall is a trait. Or being short is a trait. Hair quality is a character. Wavy hair, curly hair, straight hair, these are traits. Okay? So, what did Mendel do? So, a little bit about the experiment. So, first thing, he worked on the pea plant. Okay, the pea plant, sweet pea. Okay. So, he worked on the pea plant. So, first things, a little bit about the plant. Plants have the male or male sex organs and female sex organs in them. Now in this, mind you, I am talking about a specific category of plants called the angiosperms. Okay, that is important. So plants have both the male sex organ and the female sex organs. So it is easy to manipulate and kind of play around to decide what you want. Okay, so kind of, if I want a purple flower, which is I want to use maybe say just the ovary, and I want no interference from the pollen, so I'll remove the pollen. Or if I don't want the ovary and I want just the pollen, I will remove the ovary. So I can kind of manipulate and use like the external agents like a brush to artificially add the pollen to a certain ovary or vice versa. Okay, and you would get certain results and can observe the results according to that, and you would know this particular offspring is the result where the male contributed these qualities, the female contributed these qualities. You kind of keep the record and try to understand. That is the one thing you can do. Another thing that you understand from this is these guys are the contributors. These are your parents in this. So it is called the parental generation or the P generation. And these are the offsprings of the seeds that you get, and then you sow the seeds and you check what quality they have. So these are the offsprings and you call them the filial. Okay, like it's a Latinized word, root word, pour la fils. Okay, so filial is like the son. So the first filial, meaning the first generation immediately after the parental generation, is called the F1. Okay. Okay. Now, what were the advantages of using the pea plant? Before that, I wanted to give a brief mention. We have had many scientists all over the world. Scientists. Okay, many people working towards trying to discover new things, work on some things, come up with a conclusion. Some were lucky and were successful, The others were not so successful. We are usually studying about the person who was successful and his findings. But remember, all in all, in this, the first step is what you select as your test subject. That is a very important thing. If you were lucky enough to get the right kind of test subject for your testing, you get your results, it kind of makes for a very convenient situation where you've got a good test subject, you worked with your hypothesis or whatever plan that you have in your mind, and you got certain results, and you can make your conclusions available to the world, and you become kind of synonymous with that experiment, and they say, oh, so and so was responsible for this experiment, and he came up with this law, and so on and so forth. Okay. Similarly, Mendel, I would say, was very lucky in choosing the pea plant. Unknowingly, sometimes you just choose, maybe he chose a lot more, and maybe he got good results with this particular plant, and he proceeded more along with it. Let's talk about what are the advantages of using the pea plant. It has a lot of distinct traits meaning you can say specific, like for example, you have something which is a character for height, you can have tall, you can have short. If you have a character for color of flower, you can have purple flower, you can have white flower. Okay, so the character is again a specific quality. A trait is one kind of that quality which shows you a certain result that you conclude from it. Okay, so it also has a very short generation time, which makes for easily growing it and seeing the results pretty quickly. It produces a large number of offsprings, and you can control the mating. Like I showed you in the previous slide, you can cut off the anther which contains the pollen grains of one flower, or you can cut off the ovary of another flower, and you can decide which flower or which pollen can artificially inseminate which ovary. All right. So each pea plant, like I said, has the male and the female organs, and cross pollination can be done artificially. Okay, so how is this experiment done, and what was done in this whole process? Let's just go along with it step by step. First, you have the parental generation. Let me just go back again. 1 minute. Okay, first you have the parental generation. We call these guys true breeding. What do I mean by that? For that, I would like for you to, generally if it is my class, I ask them to talk about their pets, and then you will have a purebred and you will have a mongrel, right? So purebred would be like, say, if you have like a Labrador, so the parents will be also labs, and their parents would be labs, and their parents would have been labs. So you are very, very sure of the parentage. Okay, so basically, if the purple flowers are purple, we know that it is always had ancestors producing only purple flowers. So all in all, it's true breeding for that particular quality, right? Once again. Likewise, if it is white flowers, it has parents and ancestors always producing white flowers. Okay, so that is how you label something as true breeding. Okay, meaning the character and the trait for that character has always been that particular kind and not any other. Okay, so when you cross a purple flower, which is true breeding, with a white flower, which is also true breeding, the F1 generation are now hybrids because this particular flower has mixed parentage, right? But it all looked purple. Where did this white go? Where? Okay, let's see. Okay, then what he did in his experiment was he took two of these hybrids and crossed them. Crossed them means cross-pollinated amongst them. He manipulated the pollination amongst them. So the parents for the offsprings have been these guys. Does that make sense? Okay. So the F1 generation, he took another of the same F1 generation and a purple flower. He crossed. He noticed in his F2 generation 75% of the population were purple flowered and 25% of the population was white flowered, which means this particular quality, the trait of this white colored flower, skipped a generation but eventually was seen one generation later. Okay, but the percentages at which he observed it was 705 of them out of 1,000 flowers that he must have estimated that had grown. 705 flowers were purple. 705 purple flowered plants he could grow, and 224 white flowered plants he could grow. So he noticed that this particular feature was not only for the character of flower color with the purple flowers and white flowers. Okay, he also noticed that for the flower position, which is some would be axial, some would be terminal. Okay, for seed color, the seeds that you get, the pods that you end up getting, the seeds are either yellow color or green color. The shape of the seed could be either round and smooth or wrinkled and rough. The pod shape is nice and smooth and inflated, or it could be constricted. The pod color could be green or yellow. The stem length or the height could be tall or dwarf-like. So he found out that for all of these characters, if he used the same pattern of using purebred parents of the two different traits that he chose, okay, and then he got an F1 wherein all of them looked like the dominant trait carrying fellow. Now what I mean by that word dominant is, in this particular case, he noticed some traits were more in their numbers than the others. Okay, so at this point he calls that particular trait a dominant trait. Okay, we will go ahead later on down the slides and redefine what you would call dominant and what you wouldn't. Right now, whatever numbers of offsprings that he got, which was in more major numbers versus the minor numbers, and especially the ones that skipped the F1 generation. That's the important sentence. Meaning, for example, if he used two purebred plants which have yellow colored seeds and green colored seeds, and he crossed them, the F1 generation, all of them showed yellow color. That would mean this green is recessive and the yellow is dominant. Okay, and then when he crossed this F1 with another F1, he noticed that 75% out of the lot, 75% of them have the yellow colored trait, and 25% of them have the green colored trait. So after his experiments and the observation, he put forth a model. Okay, he said for each of the qualities or characters there are variations. Okay, so each character there are variations or there are different traits. Okay, so you can have tall and short, green and yellow, purple and white flower, and so on. For each character there are 2 copies of genes present, and 1 copy you get from one parent and the other copy you get from the other parent. When the 2 alleles, meaning the 2 sets that are present in you, are different, okay, meaning you don't have the same alleles, different, okay, in the specific case wherein if they were purebred, both the alleles would have been the same. If they were hybrids, the alleles would be different. Okay, so that's one way to look at it. One is dominant and the other is recessive. The quality which is expressed and observed in the offspring, which has 2 different qualities present in it, 2 different alleles present in it, if the quality that particular offspring shows, even if he has like a combination of 2, that is a dominant quality. Now, what he says is when there is meiosis taking place in the system, the alleles segregate and they end up in different gametes. Gametes means different sperms or different ova, alright? Okay. And then he goes on to say, supposing you have 2 alleles for each character. So this is 1 chromosome that you got maybe from your dad. This is 1 chromosome that you got from your mom. So the particular character of flower color, it might be possible that your dad gave you the quality for flower color which was in this chromosome and it is present in this particular location, and similarly the same kind of chromosome you get from your mom and the quality for flower color is specifically present in this location. Okay, so he calls this particular location the locus of that particular gene. Okay, so a little bit more in detail about this, wherein we are elucidating what could possibly be in the chromosomes. initially what we did a few slides back, generally we just see this color, we cross it, we get this outcome, and that's it. We just put it that way. Right now we are going into a little more of detail, wherein in this detail we are talking about if this is what is the color and this is the parent, this is the two sets of that allele that he has or she has got, the parent has got. Okay. So for example, if it's the parental generation and it is purebred, this purple flower, okay, has the two alleles of capital P. Mind you, it is very important to write the letters clearly to show whether they are capital or they are small case, lowercase or uppercase. Okay, so you would have the two parental generations chosen here, purebred variety for purple flowers and white flowers. In this case it is two capital P, in this case it is two small p's. Very important to write it clearly. Now the gametes that are formed during meiosis, see, at all times during meiosis your chromosome number halves, okay, which means each of your gamete will get half the number of chromosomes, each gamete. For example, each ova or each sperm will only get half the number of those chromosomes. But the reality is this guy has got two capital P's, which means both of these sperms will get only capital P because he doesn't have anything else to offer. On the same note, if this person was the egg, both of the ova will have capital P because this particular lady doesn't have anything else to offer. She only has this set of alleles and they are both same in her or in him. Likewise, for the white flowers during meiosis, the gametes are likely to be both small p's, either egg or ova, depending on whether it is the female or the male that is doing that contribution. Okay, that is immaterial in this case. So my point is, at the end of the parental generation, when there is meiosis that has taken place, this is what they can contribute 100% of their time. That's it, no other choice. And so the combination that they offer to the offspring of this union has got a capital P and a small p. But he still looks purple and he is the F1 generation. So the appearance is purple color, the genetic makeup is capital P, small p. In this case, when he or she undergoes meiosis, likely to have gametes with 50% of the time having capital P, 50% of the time having small p. Okay, so here the likelihood of giving you variation is higher because he is a hybrid. He has one of each kind of gene, alright, each kind of that allele. Now, when you cross this with an F1 generation, that means another purple flower here of the F1 generation with a capital P, small p. And what I mean by that is in the gametes, half the time this particular flower can give you capital P, half the time he can give you small p. Agreed? So when you put this in what is known as the Punnett square, so the way it is written, okay, very important. You write the sperm on top. So you have like a square that you would make. You would write down what the sperm is capable of contributing. In this case, the sperm can give me capital P or small p. And what the eggs can give me? Capital P or small p. Okay. Now, what is the material that I have drawn in each square? These are the offsprings of each fertilization which has been successful. Okay. So, in this case, this particular sperm, if it fuses with this egg, I have two capital P containing offspring. If this particular sperm fuses with this egg, I have one capital P, one small p. If this particular sperm fuses with this egg, I have one capital P, small p. If this one fuses with this one, I would have one small p and another small p. So I have something called as two capital P's, or a capital P small p, or a small p capital P, or two small p's. Yeah, but mind you, it is not written like this. Always the dominant one or the capital one is written first and then the small one. So I have two of the two capital P small p and one of two capital P's. This means this is what I have in their genes, but this is what they look like or they appear like. 3 purple and 3/4 of all the offsprings are purple, 1/4 of the offsprings are white. Okay, so another set of terms to know and understand in this case. What you can see, you can physically see, that is your phenotype. You can see one purple here, one purple here, one purple here. So that makes for 3 purple offsprings, ratio of 3 purple offsprings to 1 white offspring. All right. But genotypically, or the genetics of it, you have one capital P, small p, one capital P, one another capital P, which makes them homozygous. Or you can have one capital P, one small p, heterozygous, another one capital P, small p, heterozygous, and two small p's, homozygous again. So the ratio of the phenotype of this particular union is 3:1, meaning 3 purple to 1 white. The ratio of the genotype of this union is 1:2:1. This is called the Mendelian ratio. Okay. Now we come across another situation as to what is a test cross. Now here, for many it might seem like, why do I need to know this? Okay, so now I see certain purple flowers in the garden. Okay, what do I already know? I know purple is the dominant variety for this particular plant. Okay, I also know that white is recessive variety, which means only and only if both alleles were recessive or two small p's, I would get white flowers. There is no way I would get white flowers if I had one capital P. That's not possible. So my next question or next situation, if I have a purple flower, I know that it has got one purple gene. I want to know whether it is one capital P and small p or two capital P's. I don't know. Okay. For this, what can I do? I can cross it and find out. But with what? Something that I am absolutely sure of. What am I absolutely sure of? This guy. I'm absolutely sure that if I have a white pea plant, that means that white pea plant has both alleles, which are two small p's. So, let's see what happens in the next line. Which means this guy, I know for sure what this guy is up to. Just let me go back. Sorry. Yeah. Okay. So, in this particular situation, I know what is the genotype of this particular plant. It is two small p's. And in this case, I do not know what is its genotype. I don't know if it is two capital P's or one capital P, one small p. So, what do I do? Situation 1. I think you are two capital P's. Maybe. I predict. I think. If that is so, my two capital P's, okay, here, and my two small p's here, I cross. I will have capital P, small p. What is this? This is my homozygous recessive plant. I already know. I am assuming this one, this part of it. Okay. So one capital P, one small p, one capital P, one small p, one capital P, one small p, one capital P, one small p. Whereas if the purple plant that I have, purple flower giving plant that I have is one capital P and one small p, that would give me a result wherein these are the capital P and small p, and this is the one that I already know. Okay, I would put them, cross them, and see what is my offspring capable of giving me, what qualities my offsprings are giving me. In this case, if you notice, 50%, I'm sorry, let's just go back. 50% of my offsprings of this union is purple, 50% is white. This is only possible if there is one capital P and one small p. In this particular case, all of it is purple. This is only possible if both the alleles of this particular unknown guy is two capital P's. So, this is one way we can find out what is the genotype Okay, this is one way we can find out what is the genotype of a dominant plant, dominant character or dominant trait plant, not character, trait plant. So what should I do? I should cross it with a homozygous recessive plant. Okay. Now, so far we've been talking about Mendel's work with Mendel's work with, let me just go back. What happened? Mendel's work with one trait. Sorry, not one trait. I'm sorry. One character. Okay, and two traits. Let us see what happens if we are talking about two characters. Okay, so we have covered a monohybrid cross. Now let's work with a dihybrid cross. Do both the characters go together, or do they get shuffled separately? Okay, so Mendel worked with two qualities, and let's see what he got from that. So the two characters that he was working with is seed color and seed texture. Seed color is the character, and the two traits that he was using in his experiment was yellow color and green color. And the seed texture character that he was using smooth and round, or he was using rough or wrinkled. Okay, so in this particular case, when he started the experiment, again he started with a purebred variety for each of those characters. So what he has used is, in the parental generation, one particular plant which was producing yellow colored seed and round shape. Okay, versus he was using another purebred variety, green colored seed and wrinkled shape. Okay, so 100% of the time during meiosis, when the gametes are being made, the gametes had capital Y, capital R in them. 100% of the time, for this particular purebred variety, the gametes had small y, small r. Okay. Now, the F1 generation of the offspring of this particular union, okay, the F1 generation, now they had a combination. So one parent gave them capital Y and capital R, another parent gave them a small y and small r. So they became capital Y, capital R, small r. So this is what this particular F1 generation was. Now, first hypothesis, to say that the character of seed color and seed quality are in the same gene, meaning wherever this gene goes, this gene also would go because they are in the same chromosome. Does it make sense? So one more time, let me just draw it in a way. I also use another example for my class. Okay? So what example I use? Okay, I'm going to try with a color pencil. So here is a romper. Okay, this romper has got a top and a bottom sewn together. Agree? Together. So I have one romper which is blue colored attached to a black colored pant. Okay, just imagine this. So this is one romper. Then I have another romper which is yellow color, which is attached to a green colored pant. Now, because they are sewn together, you can only wear them together. Does it make sense? Because they are sewn together, you can only wear them together. That is what this situation is. Let's just understand that in a minute. So, my point is, the sperm from this F1 generation, when it is making these gametes, one sperm will have the capital Y and the capital R in it, kind of like the blue and the black stuck together. And the other sperm will have the small y and the small r given by the other parent in it, kind of like the yellow and the green stuck together. Yeah? Okay. Which case you would have them have their gametes spreading in this fashion, sorting in this fashion, and the eggs sorting in this fashion, which means the offsprings would be capital Y, capital R between these two guys, or capital Y, capital R, small r between these two guys, or between these two guys, capital Y, capital R, small r, or between these two guys, small r, small r. So the ratio of their distribution will be 3:1. So this means they are dependent in their assortment. They go together. Always. Is it? Is it? Supposing second hypothesis was independent, which means, let's just go back to this particular picture that I have drawn about the shorts story and everything. Let's just do that one more time. Okay, so supposing this was a top and this was another top, okay, and this was a pair of jeans and this was a pair of jeans. Okay, and I can have how many combinations when I am distributing? Now, one parent can give me, okay, capital Y. So I can have the combinations in this fashion. Okay, I can have it just the blue and the green. This is one combination. I can have the blue and the red as my combination. Okay, I can have the yellow and the green as my combination. I can have the yellow and the red as my combination. Did you see that? How many combinations that I ended up having? So I'm just putting that in perspective now. 1 minute. Supposing this is 1. I'm just numbering them. 2. This is 3. This is 4. Okay. So my point is, I can have combinations of 1 and 3, 1 and 4, 2 and 3, 2 and 4. Does it make sense? Yeah, I can have, like, I would be able to crisscross and put those dresses together to have these 4 different combinations. Same way, I would like to get back to our real genetics of this particular situation and say, if I had okay, if I had okay, so capital Y and R, small y and R together, I would be able to get how many combinations in my gametes? I can have capital Y, capital R, one combination. Capital Y, small r, another combination. Small y, capital R, small y, small r. These are the different combinations in which my process of meiosis can shuffle the genes and assort them. The meiosis that takes place, it can shuffle the genes and assort them, which means I can have all of these combinations. When is this possible? When they are present in different chromosomes, which means the character for, okay, no, let's just try a smaller, thinner color, so I'm having space to talk about this. Give me a minute, let me try. Okay, so my point is, supposing these are my mom's chromosomes, these are my dad's chromosomes, so I have mom 1 and mom 2. This is my dad 1 and dad 2, which means the quality of, one second, I would take the yellow, yeah, the quality of okay, yellow color is here, and then let me take green. Okay, so it is present here. Does that make sense? Okay, then the quality of wrinkled and round, right? So I'm going to call that again as my normal black once again. So this is round and this is wrinkled. So because they are present in different chromosomes, they are likely to go into different sperms and or different eggs, and that is why I can have 4 combinations. I hope with this my students are able to understand why they got these different combinations in this case and not so in this case. One more time, in this particular case, I'll just go back. Supposing this is my mom and dad's chromosomes that are present, okay, so I have the, okay, and then I have the, okay, and then I have the round, and then I have the wrinkled present in the same chromosome. So wherever this quality goes, this quality will go together. Wherever this quality goes, this quality will go together. That is not the case in this guy. He has a combination. They can go in any which way, any which combination and match up. Kind of like jeans and t-shirt instead of a romper which has got the top and the bottom attached, right? So going back, you can see that when they assort, they have the distribution in such a way that phenotypically, that you look at it once again, my dear class, I'm sorry, a little bit. Phenotypically, you can see that they have 9 yellow seed, okay, 9 yellow and round, okay, and then the ratio, okay, the ratio when they counted it, and then 3 green and round, 3 yellow and round, and 1 green and wrinkled. Okay, so this is Mendelian ratio for a dihybrid cross, which is 9:3:3:1, and it matched up with the real results. That is what you observe on your Punnett square. It matched up with the real results, wherein they got something like 315 of the yellow round, 108 of the green round, 101 of the yellow wrinkled, and 32 of the green wrinkled. Okay, so observation and counting confirmed that independent assortment was possible. It applies to genes that are present on different non-homologous chromosomes. The genes that are located near each other on the same chromosome tend to be inherited together. Meaning, if it's in the chromosome, if it's close together, they will go together. The reason being, during the meiosis, there is a particular stage, if you recall, called the prophase 1. Here, what you do is there is something known as crossing over, and this gives us a lot of variety. But if they are together, they tend to be crossed over together. But if they are far away from each other, this might go to one chromosome, this might stay in another chromosome. So they tend to assort as though they are independent of the other. Okay, now we move on to the topic of probability. So my point is, when you toss a coin, okay, what is the probability that you would get a heads? Okay, 50%. Meaning, if I toss 100 times, 50 times I might get a heads. 50 times I might get tails. Okay, or if I am talking about a pack of cards, you know, pack cards have 52 different cards in them. What is the probability of drawing an ace of spades? 1 by 52. Okay, so what is the probability that I would draw something other than an ace of spades? And that is 51 by 52. Okay, the probability of all the outcomes should add up to a 1. Okay, so all the different outcomes, if you add up, it should add up to become a 1. Okay, that is one. Like, for example, tossing of coins, like probability of getting a heads is 1 by 2, or probability of getting a tails is 1 by 2. Probability of all the outcomes is half plus half is 1. That is what he's talking about. And each outcome is independent of the one before or the after. What does this mean? If I'm tossing a coin, each time that I'm tossing a coin, that probability is independent of what happened before or will be independent of what is happening afterwards. It has got no no say in what is happening before or the after. Similarly, in your meiotic separation and formation of gametes, the probability of getting a certain trait in the offspring depends on the number of variants or number of alleles that you will have, different kinds of alleles, and the alleles that your lady or the mother has, and then you would put all of those probabilities, put them together and test the outcome and get a final probability of a certain trait coming in the offspring or not, observable in the offspring or not. Okay, then we have a certain set of rules called the multiplication rule and the addition rule. So the way it works is, supposing normally what I do is, in my class I ask them to toss a coin. When they toss a coin, I say, okay, what is the probability of getting a head? They will say half. Okay, and then I'll say toss a coin again. Okay, what is the probability of getting a head again? Half. Again toss a coin. What is the probability of getting a head again? Half. Now when I say, what is the probability that you toss 3 times and all 3 times you get a heads? That they will calculate by saying half into half into half, meaning they multiply each of the probabilities and the consecutive 3 times, and they say the probability that I would get a head all the 3 times is 1/8. This particular rule is called the multiplication rule. How does it go in words? The probability that 2 or more independent events will occur together is a product of their individual probabilities. The probability in an F1 monohybrid cross can be determined by using the multiplication rule. Okay? Segregation in a heterozygous plant is like a flipping of a coin. Each gamete has half the chance of carrying the dominant allele and half the chance of carrying the recessive allele. Okay, so here you would see, if this is an F1 generation, so the probability of this particular sperm having capital R is half. The sperm having small r is half. Same way the eggs having is capital R is half, and this eggs having small r is half. So the probability of the capital R fusing with the capital R of the egg, the two probabilities, that you'll have to multiply and you get a 1/4. The probability of you getting a heterozygous, in this you multiply by the 2 halves and you get your 1/4, and then you get your 1/4, and then in this case you get your probability of the 2 of them contributing small r is again the 1/4. Okay. Now, the addition rule, the probability that any one of or two, the probability that any one of 2 or more mutually exclusive events will occur is calculated by adding the sum together of their individual properties. Now, it might sound very complicated to you when you hear it. 1 second. I don't know, for some reason I am not able to. Yeah, 1 second. Okay. It is sounding very complicated, but it is not. And any one of you can solve it even without realizing that you are actually using the addition rule. For example, now think about the previous toss experiment that we did and kind of tie it in with our addition rule. This particular slide that I am going over, I am going over this to explain the addition rule. In this particular case, what I am saying is, if you are looking to get a heterozygous offspring, okay, how would you get that? If the sperm contributes capital R, the egg has to contribute small r. If the sperm contributes small r, the egg has to contribute the capital R, which means they are mutually exclusive. Now, meaning in this particular case, if you see, if the sperm is contributing the capital R, it cannot contribute the small r. Same way, if the egg is contributing the capital R, it cannot contribute the small r. The event is mutually exclusive. And so, the probability of getting a certain mutually exclusive event to occur is calculated by the addition rule. So we go back and see that. Okay. In this particular case, again, when you see, the probability of the sperm contributing the small r is half, and the egg contributing the capital R is half. So you get this heterozygous event 1/4 of the times. And the probability of the egg contributing capital R and the sperm contributing small r, you get this particular situation 1/4 of the times. Now, our question was, what is the probability that you would get a heterozygous individual? What will you say? This is 1/4, this is 1/4. You will add that up. Half you've added. In this case, that means you have used the addition rule. I hope that's clear. Okay, so one more time, the probability that any one of two or more mutually exclusive events will occur is calculated by adding the sum of their individual probabilities. Okay, so the addition rule can be used to figure out the probability of getting a F2 heterozygous or a zygote from a monohybrid cross where the dominant allele is from the sperm and the recessive allele is from the egg, and vice versa, and add the individual probabilities. Now we move on to the part where we are talking about where inheritance patterns are not really very clear cut. There isn't like I will either do this or. There is like an in-between kind of situation where you have to figure out. There isn't something which would say, yes, this is going to be a purple flower color trait or a white flower color trait, and that's it. So you might have something which is more complex, and that is what we are discussing in this future slides. Okay, so not all characters can be determined very simply. Some of them are not completely dominant or completely recessive. Sometimes some genes have more than 2 alleles. Sometimes you have multiple phenotypes. So with all of these situations, we have different kinds of inheritance patterns for different qualities. Let's go over each one of them. Okay, so you see complete dominance in pea plants. So you do see something known as a purple flower color trait or a white flower color trait. End of story. Then you have something called as incomplete dominance, which I will discuss in the next sheet. Okay. So this particular chart kind of explains the incomplete dominance. This particular flower is not the pea plant flower, please. It is called the snapdragon. It's a very pretty looking flower. They have used two colors distinctly in this case. One is white color and one is red color. So you can be red if both your alleles have the red colored quality. You can be white if both the alleles have the white colored quality. Okay, the gametes that they produce 100% of the time is the red colored trait. The gametes that this guy produces 100% of the time has got the white color quality trait. Okay, now when you have this combination fellow who is heterozygous in the F1 generation, in our purple plant we would have chosen one as the dominant and the other as the recessive. In this case, it doesn't work like that. It looks as though it is like a blend of the two, or both of them are asserting their quality. So it is giving me a combination of a pink colored variety. So the F2 generations, when you cross them, these guys have 50% of the time they can give you CR and 50% CW. Likewise for the egg, when you cross them, 1/4 of them will give me CR, because now both the alleles of this particular offspring has the CR gene, and you would have 1/4 of them giving me CW, because both the alleles of this particular offspring have CW gene. Here in this particular case, you have CR and CW as a combination, and so neither of the color is missed, neither of the color is completely expressed. This is incomplete dominance for you. Okay, we go back to talking about the codominance. Codominance is another trait that we will be looking at in a few slides from now. Okay, so codominance basically is using a cow's example, which is say a white colored cow or a beige colored cow, mostly white, let's take it, and a brown colored cow. The offsprings, all of them will be patchy, which means both genes assert themselves. This is what you end up getting called as the roan. Okay, this is codominance for you. Mind you, it is different from incomplete dominance. In incomplete dominance, you do not see any of the parental color distinctly, not seen distinctly. In codominance, you can see the parental colors, but in patches. Okay. Okay, before we talk about this particular feature once again. Okay, so here I get back to talk about multiple alleles. A little bit about multiple alleles will be clearer once I talk about what our blood groups are. So here is where I ask my class what their blood groups are, and some of them seem to know what is their blood group. So they would say, Miss, I am group O, or I am once again. So they end up saying, I am group O, or I am group A, and they'll say I'm O positive, I am A positive, or A negative, or O negative, or B positive, or B negative, or AB positive, or AB negative, and so on. Okay, so these are the different phenotypic traits that we can see. Now, is it through one particular allele or just two alleles in action? No, It is actually more than two alleles in action, which result in this kind of multifaceted combinations that we have. Okay, for that, a little bit about the blood groups itself. Okay, so each and every red blood cell has a glycoprotein on the outside. Okay, if it has a certain glycoprotein kind of like this, I would call it like, say, A. So it has blood group A. Okay, so the gene that is capable of giving this particular quality is, if I have the IA in me. Okay, so I can have IA, meaning both parents giving me this quality, or I can have IA and I, one parent giving me this quality and one parent giving me this quality. How does that happen? Let's see. So this is say, for example, I have put this is blood group B. So now I can be blood group B because I can have one parent give me IB and the other parent give me IB, and so I am blood group B. Or I can be blood group B if one parent gave me the IB and the other parent give me an I. I can be blood group A and blood group B, which is going to make me AB. How did that happen? I can have one parent give me IA and the other parent give me an IB. That's it. This is my combination I can have. Now, I can have blood group where I have neither of these groups sticking out. So I am blood group O. How did that happen? Because both my parents gave me the group smaller this. So I have three different alleles which shuffle together in the entire population and has gotten distributed amongst different people to finally represent different quality, different outcomes in different people in the human population. So the gene alleles that I am talking about is IA or IB or small I. So these together in different shuffled combinations for different people give me a final character outcome in different people. And this is why, because it is more than two alleles that are in play, it is coming under the grouping of a complex mode of inheritance called multiple alleles. I hope that's clear. Okay, then we have something known as pleiotropy. Pleiotropy meaning one gene that you end up having, or say one trait that you end up having, causes so many physically observable situations. One gene, and that is called pleiotropy. For example, the person who is homozygous for the sickle cell allele, so he ends up making an abnormal hemoglobin protein. This hemoglobin protein crystallizes and causes the red blood cells to become sickle shaped. Because of that, what happens? They have physical weakness, they have anemia because the blood is not able to supply a good amount of oxygen to all parts of the body. They have heart failure with blockages, they can have pain and fever, they can have brain damage, they can have damage to other organs, they can have spleen damage. It can also lead to kidney failure, rheumatism, pneumonia, another infection, paralysis, and impaired mental function. So you see, one gene can cause such a huge number of phenotypic effects. Or physically observable situations. Another example is cystic fibrosis. Cystic fibrosis is a situation wherein the body is kind of not able to take in the chloride and it keeps throwing the chloride out. And in this process, there is a kind of mucus that is formed outside the cell, and that kind of clogs the respiratory system or the lungs. So you would have congestion and forever having breathing problems, health issues, physical weakness. You can have trouble having physical exercises, and you can have lots of infections, like you become prone to a lot of infections. So you see, this particular situation is inability to take in the chloride. It keeps throwing the chloride out of the cell. That is the one quality which leads to so many physically observable effects. That is another example for pleiotropy. Okay, then we have something known as epistasis. Okay, so for understanding epistasis, final outcome that I have, okay, for example, my Labrador is beige colored, is because of two different genes present in two different places. They come together and give me the final expression of beige color for my Labrador. Okay, so the gene at one locus alters the phenotypic expression of the gene at the second locus. Okay, so we have an example to work with on this. Okay. So he says, in this particular case, there is a gene called E and small e. There is a gene, let me just, okay, there is a gene called E, dear, E, capital E, then there is a small e, which is the allele. Okay, then there is another one called capital B, and there is an allele. So depending on what is the combination of these genes, the dog or the offspring puppy is capable of expressing a certain color. Okay, so one of them is responsible for making of the pigment. The other one is responsible for actual placement of the pigment. Okay, so in this case, if I have at least one capital E and one capital B, that particular lab will be black. So you see, one capital E and one capital B, black. One capital E and one capital B, black. One capital E and one capital B, black. So you would have one capital E and one capital B, black. Likewise here, black. Likewise here, black. Likewise here, black. Likewise here, black. With respect to my chocolate, the one capital E and it can be two small b's. It has to be. If it was one capital B there, it will become black. So one capital E but two small b's, and it would end up being brown or chocolate in color. So capital B but two small b's, capital E but two small b's. So chocolate colored lab, chocolate coated lab. Okay. Third option, the e's, if they are two small e's, no matter what you have in the B, it doesn't matter. If it is two small e's, you will have a lab that would be beige in color, meaning two of the different genes have come together to decide the final outcome, phenotypic outcome of a certain character. Same example but with a different animal. Again, in this particular case, there is capital B, small b, capital C, small c. If I have at least one capital B and one capital C, it will be a black colored coated mouse. 1, 2, 3, 4, 5, 6, 7, 8, if you look at all of them. Then the second one, if I have at least one capital C, at least one capital C in this case, it will be a brown coated, but it has to be two small b's in this case, like the previous one. This guy has to be two small b's and at least one, it will be a brown. And finally, it has to be two small c's. So no matter what this guy is, it will be a white colored mouse. Yeah. Okay. So these two small c's decide the final coat placement. If they are recessive, no matter what pigment I produce, I cannot place it in that coat. Okay. So that is epistasis for you. Now we move on to another variation wherein it is extremely difficult to explain how a human being inherits certain qualities, because it is not dependent on just one allele or just two alleles. It's a range of different multiple alleles working on different combinations to give you a continuum, color in a continuum or in a range. So what I would like for you students to appreciate in this is, for human skin color, he is using gene 1, gene 2 and gene 3. Okay, these three genes they use, and you would have them, say for example, called as D1, D2 and D3. Okay, and they are expressed in different ranges, and that would explain whether a person has how many number of dark skin genes. Okay, 0 number would mean you are very light in skin tone. 6 numbers will mean you are very dark in skin tone. The next page will help understand it clearer because it has got the blocks of color put in different squares. So this probably runs home the point of what we were talking about. So we had something called as D1, D2 and D3 in different combinations, and that kind of sort of explained how many of the skin color genes you would have. None, you are light skinned. All of them, you are dark skinned, right? So you can have them in different combinations possible. Okay, that is why it is called polygenic inheritance, where the inheritance is in a continuum. Similar to this particular situation is human height. Okay, it also goes through a range. There is people with different height ranges that exist, and it is also decided by a host of different genes that come together in different combinations to finally decide the outcome. Okay, we move to the part where I'm talking about environmental impact on the phenotype, meaning you would have the phenotype of a character which will depend on the genotype, meaning what is present in your genes. It is also dependent on the environment. An example is hydrangea flowers of the same genotype can have blue, violet to pink color depending on the pH of the soil. So, ideally, if you have more acidic soil, even if it has the same set of genes, the flowers would be pink in color. If you have more basic soil, the flowers would be more blue in color. So like I tell my students that if you go to any nursery, the soil that they have grown these hydrangeas, they would have added certain chemicals to make the soil more acidic or basic in order to get the color, and it might not, that same flower might not give you the same color once you take it home and plant it into the ground, and if you have not given it the same set of chemicals. Interesting. So you can also have other diseases which or disorders which have so many different factors which decide the outcome, meaning heart disease. You can have more genetic and environmental components deciding on what is the outcome, or say cancer. Okay, that a person should suffer from a heart disease or from cancer is maybe some part of it is genetic, the other part of it could be diet, could be stress, could be lifestyle, etc. Okay, humans are not very good subjects for genetic research because the generation time is too long. Okay, and the parents produce very few offsprings. Breeding experiments that we do are not in plants and in animals is not really ethical or acceptable to be conducted in humans. So we are still lagging behind in human genetic research as compared to the amount of genetic research that can be done in plants and in animals. But it is heartening to know that the basic Mendelian genetics is the foundation for human genetics also, like we'll see in the next slide. Okay, so doing a pedigree analysis. A pedigree is a family tree that describes the interrelationships of parents and children across generations. So inheritance patterns of particular traits can be traced and described using pedigrees. So you can look at two different situations. One having a widow's peak and one not having a widow's peak. One that can roll the tongue and one that cannot roll the tongue and one can. One that is having a whichever thumb on top. One that has got the length of the second toe shorter than the big toe or longer. One that has got front teeth that are closed together or there is a definite gap. Then you can have attached earlobe or free hanging earlobe. You can have the little finger that is straight or crooked. Okay, so these are different characters for which we have run some pedigree analysis and found out which one is dominant, which one is recessive, and which one have you inherited from your parents, or which one are you likely to inherit because you look at the parentage and so on. For example, in this particular case, okay, so here are two sisters. Okay, so two sisters, one of them has a widow's peak and the other doesn't, which means definitely parents, one has and the other does not. So these two sisters are a combination of a set of parents who have widowspeak and one has and one doesn't. So going back here, so a lady who has widowspeak, it is already known that having a widowspeak is a dominant trait. So if you have one allele, one capital W, you are likely to express it. And you have to have two small ones in order for you to have no widowspeak. But this particular lady, is she heterozygous or homozygous? Now, to understand that, we have to go back and look at the parents. Definitely, if they have one recessive traded child which has got two small w's, that means one has come from each parent. That means the parents are definitely this. Is that so? The both parents have the widowspeak, but one child has it and the other doesn't. So this particular child is either a combination of a capital W and a small w, or two capital W's. Okay, that is what her inheritance patterns could be. And then we go back to look at the ancestry, wherein we look at all the sisters, siblings, family, and kind of trace it down to which parent got it, which parent doesn't, and so on and so forth. So you can trace a certain trait along the different generations, across the different generations, by charting out a family tree and discovering from which parent or from which grandparent you would have inherited such a trait or not. Same way you would have attached earlobe and a free earlobe. That is also another trait that can be traced back. Now, it seems very frivolous to be talking about genetics and covering things like widowspeak or free earlobe and attached earlobe. Mind you, it can also be put to use when you're trying to trace genetic diseases, to find out the inheritance patterns there, or from where you have ended up getting, or from which person in the family has this particular situation and from whom it must have been like a hand-me-down. Okay, so it can be used also to make predictions about the future offspring. Like we kind of can take an estimate or a guess to find out if you are homozygous for a certain trait or a heterozygous for a certain trait, and likewise for your husband or your spouse, and then kind of find out what will be the possible situation for the offsprings. So you can make predictions for the future offsprings. You can also use multiplication rule, addition rule to predict the probability of certain specific phenotypes. Okay, then now we are going to be talking about certain recessive alleles. Recessive alleles means both the alleles should be recessive in order for you to express it. Okay, so many genetic disorders are recessively inherited. They still exist in the population because there are carriers, meaning there are many people amongst the population who would be heterozygous for it, in which case they have one dominant allele and one recessive allele, in which case this doesn't get expressed but this gets transmitted to the next generation and so on. Okay, one example that we can study is albinism. When do you see the expression of any recessive trait is more? Okay, is when there is inbreeding, when there is captivity, when there is consanguinity. Consanguinity means mixing of blood, meaning incestuous outcomes, with something that is taboo, something that customs prevent or encourage. For example, in ancient Egypt, they were allowed to marry the sisters in order to keep the line purebred. Another example for such in recessive alleles is Tay-Sachs. I can explain that one more time when we go to the next slide. So here we have two normal parents. Okay, the genetic disorder of albinism is inability for the pigment to get deposited, okay, of whatever pigment is being made to get deposited, and so the affected offspring would be kind of very white with no pigment at all in the skin, and you will see how it works. So you would have normal parents who contributed 50% of their time a normal gene and the 50% of their time an albino gene. Same likewise, if two normally or physically normal looking parents came together, they probably or may be carrying their recessive gene, which means both parents are carriers. Okay, when they carry that, 3/4 of the time they would have normal looking offspring, of which 50% of their offsprings would be carriers. 1/4 of the time they can end up having an albino offspring. Okay. So other recessive autosomal. Now what I mean by autosomal in this particular case is not present in the sex chromosomes. It is present in the other 44 chromosomes and not in the 2 chromosomes which are the X or the Y or the 2 X's. These are your autosomes. So recessive autosomal would mean it is not specific to a certain sex of a person. It can be in the autosome of any person, boy or girl, male or female. Cystic fibrosis, sickle cell anemia, all of these are recessive autosomal disorders. Then there are some dominantly inherited autosomal disorders. So remember, dominantly inherited disorders, even if you have one allele, you will express it. That is sad. There can be some that are lethal, and so if they are lethal, we would lose the offspring very quickly, and you would not see it still being present in the population. There is, however, one of them which is lethal, but unfortunately, we do not realize the onset of this particular disease till we reach age 30. By then, we may have married, had children, etc. So, other dominantly inherited autosomal disorders are polydactyly, achondroplasia. Polydactyly is many fingers. That's what it means. but it actually means one extra finger. Achondroplasia is dwarfism. Okay, so most of the recessive autosomal diseases are commonly present in the population, but the chances of an individual having both the recessive genes is low, and chances of having carriers is high. In the dominantly affected autosomal disorders, they are not necessarily very high, because if it is dominantly inherited and if it is lethal, you wouldn't have that gene being passed over to the next generation, because the individual dies before it's being passed to the next generation. So a little bit about achondroplasia. This is normal person for this particular gene has two recessive alleles. So the general population has, all over the world, okay, has recessive alleles for this particular gene. Even if they have one allele, they will end up expressing it. That is the beauty of dominant inheritance. Okay, so your capital D and small d present in the one parent and two small d's present in the other parent might have two normal children and two dwarf children in the distribution. Okay, so genetic counseling can help. Now, what are the different kinds of genetic testing that is possible in this modern day and age? Okay, first, what do genetic counselors do? They can provide information to prospective parents who are concerned about the family history for a specific disease and counsel them based on Mendelian genetics and probability rules. This helps the couple determine the odds that their children will have genetic disorders. But the downside is, if insurance companies can access this information, they can say that it is one of those preconditions, and so they might refuse to give them insurance, or it can prejudice some companies to give people jobs, because they might end up having to shell a lot of medical insurance money to such people. Okay, so for identifying the carriers, there are many tests, and it helps define the odds more accurately. You can do fetal testing or newborn screening. In fetal testing, you can have two types: invasive or non-invasive. Amniocentesis, the liquid that bathes the fetus, is removed and tested for presence of certain chemicals and also for karyotyping. Once that is done, they kind of look at it and then make their conclusion about what is the genetics of the baby that is conceived. Okay, and in chorionic villus sampling, a sample of the placenta is removed and tested for certain chemicals and karyotyped. Now, you can also have techniques such as ultrasound, wherein you can have a non-invasive method of looking at the development and checking for presence of certain genetic disorders like spina bifida, okay, or fetoscopy, that can allow for fetal health to be assessed visually in utero. Okay, so in the amniocentesis, the amniotic fluid is withdrawn, this is the region that they would take the liquid from, okay, and they would test it for certain biochemicals presence, and also take the fetal cells, grow it and test it. It can be done. Or they could karyotype from the chromosomes that they extract and place it on the computer and kind of align everything in place. You can also do chorionic villus sampling, wherein you can start this even when earlier than like within several hours you can get results. This takes a longer time to give results, and this takes a longer time to give results. Chorionic villus sampling is to take a little bit of the placenta through a suction tube and take the fetal cells and estimate it for certain biochemicals, and also karyotype it to give you an idea of the genetics of the baby. Okay. And that completes this chapter, and I hope it was helpful.